Hello.
We are Group 3B and we will be presenting the various sterilization indicators for two sterilization procedures. The first is dry heat sterilization. The second, well, you must be wondering why we have included minion pictures in our presentation. Do you know what minions love most? Bananas! That's right. To quicken the ripening of bananas, a chemical substance called ethylene is used. In the second part of this presentation, we will be going through a type of sterilization involving a related compound, ethylene oxide. First, we will start by covering the sterilization indicators for dry heat. Dry heat sterilization is the use of moisture-free hot air to kill microorganisms by oxidation of cell components. It is used for depyrogenation and as a method of sterilization. It may be performed in a hot air oven or a hot air tunnel. There are three types of indicators for dry heat. Physical and chemical indicators are used to monitor temperature, which is the most important physical parameter for dry heat. Biological indicators, which use the spore content and endotoxin, are not usually used. Products can be released on the basis of physical process data rather than on the basis of biological indicator data, as the process parameters are already understood.
Thermocouples are used to measure temperature as they are able to withstand the extreme conditions within the sterilizer.
Thermocouples are used to measure heat distribution and heat penetration. To measure heat distribution, thermocouples are placed outside the load and at various parts of the sterilizer, as represented by the blue bars. At least one thermocouple should be placed at the coolest part of the sterilizer to ensure that it reaches the desired temperature. To measure heat penetration, thermocouples are inserted into the load to detect the product temperature, and this is represented by the red bar. During the sterilization process, the temperature is taken with each sterilization cycle and recorded in a chart.
Thermocouples should possess these properties. They must be durable for repeated use as temperature indicators in steam sterilization, validation, and monitoring. Copper Constantan wires coated with Teflon are a popular choice as thermocouple monitors. The accuracy of thermocouples should be within 0.5 degrees Celsius. With time and use, degradation of the thermocouple and its calibration is inevitable. In the long term, therefore, a scheme of regular checks and eventual replacement should be established and maintained. When the calibration is carried out, it should be ensured that effects due to influences such as electromagnetic interference or mechanical stress are minimized. Another physical parameter is the air particulate count, which should follow the Class 100 air classification. This means that the number of particles that are more than 0.5 micrometer per cubic feet should be no more than 100. This parameter can be monitored using an air particulate counter. The second type of indicator for temperature in dry heat sterilization are the chemical indicators. They undergo changes in color or physical properties after being exposed to sterilization temperatures and serve as a final check. However, they cannot prove sterility in a product. Recall that Brown's tube is a small glass tube containing a heat-sensitive dye. For dry heat sterilization, Brown's tube type 3 is used. At a temperature of 160 degrees Celsius, there is a change of color from red to green. Another chemical indicator is the witness tube. This is a sealed glass tube that contains a crystalline compound, which melts at the sterilization temperature. A dye may be added into the tube to show more clearly that the crystals have melted. There are a number of compounds that can be placed in the witness tube with melting points that correspond to the sterilization temperature. For example, at 115 degrees Celsius, sulfur can be used. At 122.5 degrees Celsius, benzoic acid can be used. Dry heat indicator labels are another chemical indicator of sterilization. These are adhesive labels that can easily be applied on the products to be sterilized. They are printed with a chemical indicator that will change from green to brown when exposed to temperatures at 160 degrees Celsius after a period of 10 minutes. This color change is irreversible and thus it will be apparent if the products have been exposed to the sterilization temperature or not.
The biological indicators are used to measure the spore count and endotoxin levels after dry heat sterilization. Spore suspensions and spore strips are used to measure spore count. On the other hand, the Limulus amebocyte lysate LAL test is used to measure endotoxin level. However, it must be noted that biological indicators are not necessary if overkill cycles are used. To assess the effectiveness of the sterilization procedure, standardized bacterial spore preparations are subject to dry heat sterilization and then aseptically transferred to an appropriate nutrient medium to test for the total viable spore count. The spores of Bacillus atrophaeus are most commonly used. The spore preparations can come in the form of spore suspensions or spore strips. According to the BP, the number of viable spores must exceed 1 million per carrier, and the D value at 160 degrees Celsius should be more than 2.5 minutes. According to the USP, viable spore count is determined using the pour plate method. The recovery medium used is 20 milliliters of soybean casein digest agar medium. The number of colonies is taken to be the number of spores that remain viable after the dry heat sterilization. The number of spores should show a 10 to the power of 6 reduction. If dry heat sterilization is used for depyrogenation, the endotoxin level should be reduced to at least 10 to the power of -3 of the original amount. The Limulus amebocyte lysate LAL test can be used to measure the amount of endotoxin. LAL contains clotting factors obtained from lysing the white blood cells of the horseshoe crab. The presence of endotoxin activates the clotting cascade and results in the formation of insoluble coagulant clot. There are 3 LAL test methods: the gel clot, turbidimetric, and colorimetric methods. For all the methods, the concentration of the unknown sample can be determined from the standard curve. The gel clot method involves the formation of a gel. In order to test the endotoxin content of a sample, a small amount of the sample solution is added to a test tube containing the LAL powder. The test tube is then incubated at 37 degrees Celsius for 45 minutes. After incubation, the tube is inverted by 180 degrees in one smooth motion. If the tube contents remained upon inversion, it means that a firm gel formed and a positive result is obtained. Conversely, a negative result is obtained if an intact gel is not formed. The second method is the turbidimetric method. As the concentration of the endotoxin increases, the amount of insoluble coagulant increases, thereby increasing the turbidity of the mixture. A spectrophotometer is used to measure turbidity to determine the concentration of endotoxin. For the colorimetric method, a synthetic chromogenic peptide susceptible to hydrolysis by the LAL clotting enzyme is mixed with the LAL reagent. This synthetic peptide contains a chromogenic tag, usually para-nitroaniline, which is released free into the solution by the clotting enzyme. This will produce a color change from colorless to yellow. The intensity of the color is measured through spectrophotometric analysis of the test sample at 405 to 410 nanometers. Now we are going to discuss the sterilization indicators for ethylene oxide sterilization, also known as ETO. This is an overview of the different indicators for ethylene oxide sterilization. The essential physical parameters for effective ETO sterilization include gas concentration, temperature, and relative humidity.
There are 3 critical physical parameters in ethylene oxide sterilization that has to be monitored for each cycle. They are temperature, concentration of ethylene oxide, and relative humidity. The first indicator that I will go through is thermocouple for temperature. Ethylene oxide sterilizer should have their thermocouple sensor placed in the jacket and chamber. Temperature sensors should be located in areas that are most likely to represent the lowest and highest temperature, such as the unheated area of the chamber and areas near gas entry where temperature will be higher, similar to dry heat sterilization. Ethylene oxide concentration and relative humidity can be measured by either direct or indirect methods. Both parameters can be measured directly using gas chromatography and infrared spectroscopy. For gas-liquid chromatography, it separates the gaseous components based on differences in their solubilities in the liquid stationary phase. Because ethylene oxide and water vapor have different solubilities, they have different separating rates. In infrared analysis, the absorbance of the gaseous components are measured at a particular wavelength. The graph on the right shows this. The black line is ethylene oxide, while the red line is water vapor, which is also a measure of relative humidity. You can see that the major absorbance peaks of ethylene oxide and water do not interfere significantly with each other, making this method viable. The pots where samples are taken should be properly located to obtain a representative sample of an ideally homogeneous gas mixture.
Ethylene oxide concentration may be indirectly obtained through calculations. The first method is based on pressure and temperature, using the ideal gas law equation as shown. The second method is based on the weight of ethylene oxide introduced into the sterilizer and the volume of the chamber. Because commercially available ethylene oxide are usually diluted, there is a need to specify the ethylene oxide fraction. This calculation assumes no leakage, polymerization, or liquefaction. But if they were to occur, the concentration calculated will be higher than expected.
To obtain relative humidity measurements indirectly, calculations can be done using the equation shown. Partial pressure of water vapor is determined from the change in absolute pressure of the chamber after injection of steam. The saturated water vapor at a particular temperature can be found in reference text. Indirect methods also can be used as a backup or for cross-checking to determine if sensors involved in direct measurements are faulty.
Next, we will be discussing chemical indicators in ethylene oxide sterilization. The most common chemical indicator used is sterilization indicator tapes or labels. Under adequate exposure of sterilization conditions, the tapes will change from violet to green. Color change indicates that the product has been exposed to ETO sterilization process, but cannot prove that the product is sterile. The violet to green color transition is sensitive to time, temperature, humidity, and presence of ETO. Chemical indicators should be used in conjunction with biological indicators, but should not replace them because only a biological indicator composed of heat-resistant spores can measure the microbial killing power of the sterilization process. It is a convenient and inexpensive method to distinguish the items that have undergone sterilization. The Royce-Bowler test involves the use of a polythene sachet of specific dimensions containing magnesium chloride, hydrochloric acid, and bromophenol blue indicator. As ethylene oxide is slowly diffused in, a reaction occurs, forming ethylene chlorohydrin and a color change of yellow to purple. Time taken for the color to change is based on temperature and concentration, and it will correspond closely to the time taken to sterilize a product. Moving on, we will be discussing two different biological indicators, which are paper carriers and attest tubes. In gaseous sterilization, the relevant process parameters are not always clearly understood. Since the compliance to physical parameters is insufficient to conclude sterility, biological indicators are especially important in ensuring sterility of the sterilized product in every cycle. Some preparations of B. atrophaeus are commonly used. Paper carriers are filter paper inoculated with spores and can be in the form of strips or discs. The strips are packaged in a peel-open paper pouch. The paper pouch is permeable to ETO but resistant to moisture and air at ambient temperature or pressure. One paper carrier is used as a control, while others are sterilized with the load. They are placed at chamber locations where sterilization is the most difficult. After sterilization, the paper carrier is incubated in nutrient broth at 30 to 35 degrees Celsius for 7 days. The sterilization process is unsuccessful if there is turbidity in the tube, which implies bacterial growth. It is important to note that the results are only valid if the control strip shows growth. Another biological indicator is the attest tube. It consists of the following components. The gas permeable cap allows ETO to pass through freely to interact with the paper carrier at the bottom. When a test tube is used as a control, while at least 2 other tubes are sterilized together with the load. After sterilization, the test tubes are incubated in a vertical position at 30 to 35 degrees Celsius for 48 hours. If there are microorganisms present even after sterilization, acid production associated with growth will cause the color of the medium to turn yellow. As with the paper carriers, the results are only valid if the control shows growth.
In conclusion, temperature is the most important parameter to monitor for dry heat sterilization, and thus there is a greater emphasis on physical and chemical indicators to demonstrate that the temperature in the sterilizer is sufficiently high for the correct amount of time. In contrast, biological indicators are more reliable for ETO sterilization because relevant process parameters are not always clearly understood. Carrying out a biological test would be more conclusive in determining if sterilization is performed appropriately. We have now come to the end of our presentation. We hope you have learned more about the different indicators for dry heat and ethylene oxide sterilization. Thank you.